Welcome to the class. Today I am going to teach you about the biological and the nutritional questionnaire. In before videos I have covered the subjective and the objective classification and types of methods for food analysis. Now coming to the topic, biological methods of food analysis. These are the techniques to detect, identify and to measure the various components, contaminants and the microorganisms in the food products. Why do we use these biological methods for food analysis? It is useful for food safety, detecting pathogens and the presence of specific proteins, and DNA sequences are assessed. Here are some key biological methods used in food analysis. One is microbiological analysis. It is used to detect and quantify microorganisms in food, including bacteria, yeast, molds, viruses. It is critical for assessing food safety and food quality. Now coming to the plate count method, it involves culturing microorganisms on nutrient agar plates and counting colony forming units to estimate the microbial load. This plate count method is used to estimate the number of living microorganisms in a sample. It is applied to count the bacteria and the fungal load in the food, water and environmental samples. It is a microbiological technique. It is used to estimate the number of microorganisms in the sample. How we do this? First we will dilute the sample and we have to take the small volume of the sample into the agar plate, and we have to incubate this and allow the microorganisms to grow into the visible colonies. After incubation, the colonies are counted and the microbial concentration is calculated in terms of colony forming units per milliliter, CFU per ml. And this method is widely used in food safety, water quality testing and pharmaceutical industry to measure the microbial load. It is simple and effective for detecting the living organisms, though it is only counts those capable of growing under the provided conditions. This choice of medium depends on the type of organism being counted. That is, nutrient agar gel for general bacteria and potato dextrose agar for fungi. Next is the membrane filtration. It is used for the water and the liquid samples. Microorganisms are filtered and cultured on the selective media. This is used to detect and quantify microorganisms in the liquid samples. A measured volume of the liquid is passed through the membrane filter, which is having a pore size of 0.45 micrometer that traps the microorganisms. That filter is then placed on a agar plate containing the grown medium and incubated at a suitable temperature. After incubation, visible colonies that develop on the membrane are counted. This method is widely used in water testing and quality control in the food and the pharmaceutical industries. It is ideal for detecting the low microbial concentration, allows large volume to be tested and provides reliable results. However, it requires specific equipment and only counts viable organisms that grow on the selective media. Most probable number, MPN, a statistical method used to estimate the number of viable microorganisms in a sample. This is used to estimate the concentration of the viable microorganisms in a liquid sample. It involves inoculating a series of test tubes with a serial dilution of the sample and observing for microbial growth. After incubation, the pattern of positive and negative tubes is then compared to an MPN table to estimate the microbial concentration. This method is commonly used for detecting the coliform bacteria in water. It is useful for samples with low bacterial counts and turbid liquids. Results are an estimate, not an exact count. It is widely applied in water quality testing and food safety assessments. Next is the selective media and enrichment cultures. Use specific media and growth conditions to isolate and identify specific microorganisms such as Salmonella or Listeria. What is selective media? It is designed to favor only the growth of specific microorganisms. Remaining everything will be killed or inhibited. using the selective agents like antibiotics, dyes or specific nutrients. Example, mannitol salt agar, which selects for Staphylococcus species. Only that species will be alive in that culture, remaining everything will be killed. Enrichment cultures. These are used to enhance the growth of particular microorganism in a mixed culture, especially when they are present in low numbers. Enrichment media contain specific nutrients or environmental condition, example pH or oxygen levels, which promotes the growth of the desired organism without necessarily inhibiting others. These both methods are vital in isolating and studying specific bacteria from samples with mixed microbial population, particularly in clinical, environmental and the food microbiology. Next is the immunological methods. These uses antibodies to detect and quantify specific proteins, toxins or microorganisms in the food samples. What is enzyme linked immunosorbent assay, ELISA? A widely used technique that employs antibodies linked to enzymes to detect specific antigens, commonly used for allergens, toxins and pathogens. This ELISA is a widely used laboratory test which is used to detect the presence of the antibody or antigens in a sample. It works by immobilizing the antigen or the antibody on a plate and then adding a specific antibody or antigen linked to an enzyme. When a substrate is added, the enzyme reacts to produce a color change, which is measured to indicate the presence and concentration of the target molecule. This ELISA is also used in food safety testing and immunological research. It's a quick, cost-effective and reliable method, though false positives or negatives can occur without proper control. Lateral flow immunoassay, LFIA. We can easily understand this. Why? Because it is similar to the home pregnancy test. It is a rapid test format used for detecting allergens, toxins and the pathogens. This is a simple rapid diagnostic test used to detect the presence of a target substance, that is antigen or antibody, in a liquid sample. Liquid sample means that is blood, saliva or the urine. The sample is applied in a test strip. This is the test strip, where it migrates via the capillary action through different zones. If the target substance is present, it binds to specific antibodies on the strip, resulting in a visible lines. We can see the visible lines here, often appearing as a colored bands. This is the home pregnancy test. This lateral flow immunoassay is widely used in a point of care diagnostics like home pregnancy test or COVID-19 rapid test. It's portable, inexpensive and provides results within minutes without the need for complex equipments. Although convenient, the sensitivity and accuracy may be lower than the laboratory methods. Western blotting. This method is used to detect specific proteins in a sample after separation by gel electrophoresis. This method is widely used in molecular biology and the medical diagnostics for confirming the presence of proteins associated with the diseases like HIV or the Lyme disease. It is sensitive and specific but requires multiple steps and precise conditions, making it more time consuming than some other assays. Next is the immunomagnetic separation. This uses magnetic beads coated with the antibodies to isolate specific microorganisms or proteins from a sample. When a sample is mixed with these magnetic beads, the antibodies on the beads specially bind to the target cells or molecules. A magnetic field is then applied to pull these beads bound targets away from the rest of the sample, allowing for their isolation and separation. This method is commonly used in microbiology, cell biology and immunology for the detection or enrichment of pathogens, specific cell types or biomolecules. This IMS is highly specific, efficient and quick, making it useful for clinical diagnostics, food safety testings and research. However, it requires precise antibodies and can sometimes result in non-specific binding. Next, coming to the molecular methods. Molecular techniques involves the analysis of DNA, RNA or proteins to identify and quantify specific components in the food. Polymerase chain reaction (PCR) amplify specific DNA sequences, enabling the detection of microorganisms, allergens, genetically modified organisms and species identification. PCR is a molecular biology technique used to amplify specific DNA sequence, making millions of copies from a simple small sample. The process involves three main steps, that is denaturation, where the double-stranded DNA is heated to separate into two single strands. Next is annealing, where short DNA primers bind to the target sequences. Next is the extension, where a DNA polymerase enzyme synthesizes new DNA strand by adding nucleotide complementary to the template strand. And this PCR is highly sensitive, allowing for detection of minute amounts of DNA, and it's widely used in the medical diagnostic, forensic science and genetic research. And various of PCR is also there, that is quantitative and the reverse transcription PCR, which is for real-time monitoring and for analysis of RNA. This technique is rapid and efficient, though it requires careful optimization of conditions to avoid contamination and ensure specificity. This PCR helps to ensure the food safety by identifying the harmful bacteria like Salmonella, E. coli or Listeria in a fast and a sensitive manner. This quantitative PCR provides quantification of target DNA sequences useful for determining the concentration of specific organisms or genes. What is this quantitative PCR? It is also known as real-time PCR. It is an advanced version of the standard PCR technique that allows the quantitative measurement of the DNA or the RNA in a sample. It involves the same three main steps of PCR, that is denaturation, annealing and extension, but with the addition of the fluorescent dyes or probes that emit a signal proportional to the amount of DNA being amplified. As the PCR progresses, the fluorescence intensity increases in the real time, allowing for the monitoring of the amplification process without needing to run a separate gel electrophoresis. It also quantifies gene expression levels, detect pathogen load and assess genetic variation. This is highly sensitive and specific, making it a powerful tool in research clinical diagnostic. The results are typically presented as a cycle threshold CT value. Next is the reverse transcription PCR. It is a laboratory technique. It is used to convert the RNA into complementary DNA and then amplifying the complementary DNA for analysis. This process begins with the reverse transcription, where the enzyme reverse transcriptase synthesizes the complementary DNA from the RNA templates. This is followed by conventional PCR amplification of the complementary DNA. This RT-PCR is widely used for studying gene expression, as it allows researchers to measure the mRNA levels in a sample, indicating how active specific genes are under various conditions. This technique is particularly valuable in virology for detecting and quantifying the RNA viruses such as SARS-CoV-2, the virus which is responsible for the COVID-19. Next is DNA sequencing. Determines the exact sequence of nucleotides in DNA, useful for species identification, genetic modification detection and the pathogen identification. Actually, it is a process of determining the precise order of nucleotides. We know that nucleotides are A, T, C, G in a DNA molecule. This technology allows researchers to analyze the genetic information, identify the mutations and to study the gene function of it. There are various methods of DNA sequencing like Sanger sequencing, next generation sequencing, which provides the accurate results. This DNA sequencing is widely used in genomics, medical diagnostic and evolutionary biology, forensic sciences. DNA sequencing. It is used in the food industry to identify the species and verify food authenticity and to detect the food fraud by analyzing the genetic material of the ingredients. It helps to ensure the correct labeling of the products and preventing the misidentification of the species in fish, meat and the plant-based foods. DNA sequencing is also employed to detect the genetically modified organisms and monitor the presence of allergens or the harmful pathogens. This DNA sequencing technology enhances traceability throughout the food supply chain and supporting the food safety and regulatory compliance. Its precision is critical for maintaining transparency and consumers' trust in food products. Next is the DNA barcoding. Uses short genetic sequences from a standardized region of the genomes for species identification. This is a technique. It is used to identify the species by analyzing a short standardized region of the DNA, often from the mitochondrial gene COI, that is cytochrome C oxidase 1 in animals. This unique DNA sequence serves as a barcode that can distinguish one species from the another. This DNA barcoding is widely used in the biodiversity studies, species identification and conversion efforts. It helps scientists rapidly and accurately classify organisms even from the small or the damaged samples. This method is valuable for detecting new species and monitoring environmental changes. Next is the next generation sequencing, NGS, a high throughput method for sequencing large amount of DNA useful for comprehensive microbial community analysis, metagenomics. Unlike traditional Sanger sequencing, this NGS sequences one fragment at a time. This can simultaneously sequence millions of fragments, providing vast amount of data in a single run. The process involves preparing a library of DNA or RNA fragments followed by the amplification and sequencing using various platforms such as Iron Torrent or PacBio. This NGS is widely used in genomics. It's a crucial tool for understanding the genetic diseases, developing personalized medicine and exploring the biodiversity. Next is the biochemical methods. These biochemical tests are used to identify microorganisms or assess metabolic activities based on the enzymatic reactions or other biochemical properties. What is biochemical kits or panels? Use a series of biochemical tests to identify bacteria and fungi based on the metabolic activities. These biochemical kits are the laboratory tools used to perform specific biochemical tests for diagnosing diseases or monitoring health. These kits contains reagents, standards and control necessary for testing various parameters such as glucose, cholesterol, liver enzymes, etc. This panels involves typically group multiple tests for comprehensive analysis, allowing for the assessment of the patient's metabolic status or the organ function in one procedure. These biochemical kits used to analyze the nutritional content, detect contaminants and ensures the safety and the quality of the food products. These kits can test the components such as sugars, proteins, enzymes and fats as well as the harmful substances like toxins, allergens and the pathogens. Their quick and reliable results are widely used in routine quality control, food safety monitoring. Next is enzyme assay. Measure the activity of specific enzyme to assess the presence of certain microorganisms or the biochemical process. They also provide the valuable information on enzyme function and metabolic pathway in health and disease. Next topic is the biosensors. Biosensors use biological recognition elements such as enzymes, antibodies or nucleic acids coupled with the transducer to detect the specific analytes. Electrochemical biosensors measures electrical signals generated by the biochemical reactions. This is nothing but the analytical devices that combine the biological components such as enzymes or antibodies with an electrochemical sensors to detect and quantify the specific analytes. They work by measuring the changes in the current, voltage in response to the binding of the target molecules, leading to the measurable signal. These biosensors are widely used in the food safety due to their sensitivity or the specificity and rapid response times. They can detect a variety of substances including the glucose, pathogens and toxins. These electrochemical biosensors are valued for portability, ease of use and real-time monitoring. This electrochemical biosensor in the food industry are used for the rapid and the sensitive detection of contaminants, pathogens and the foodborne toxins. These sensors works by converting the biological interactions such as the binding of specific molecules, bacteria or the chemicals into an electrical signal. They are commonly employed to monitor food quality, detect pesticide residues and ensure the food safety by identifying spoilage microorganisms. Electrochemical biosensors are portable, cost effective. Their use improves the efficiency of food safety monitoring and helps to maintain the regulatory standards. Optical biosensor detects changes in the optical properties such as fluorescence or absorbance due to the analyte binding. These are the analytical devices that utilizes light-based detection methods to identify and quantify biological methods or pathogens. They typically involve the interaction of light with a biological component such as antibodies or nucleic acid something, leading to the changes in the optical properties, that is absorbance, fluorescence. These are widely used in the medical diagnostic methods, food safety due to their high sensitivity and the specificity. They can detect various targets including the proteins, nucleic acids and other molecules. Next is piezoelectric biosensors, which detects the changes in the mass or acoustic waves caused by the analyte interaction. What is piezo? In Greek it is called as press or squeeze. And this is an analytical device that uses a piezoelectric materials to detect biological interaction by measuring changes in the mass or the mechanical stress. These piezoelectric biosensors are highly sensitive and can detect the small amounts of bacteria or pesticides or allergens in the food products. Next is bioassays. Use living organisms or cells to measure the potency or concentration of substances. It often used for detecting toxins or the nutrient availability. Cell culture assay. What do you mean by this assay? Assay is nothing but the test or the investigation or the analytical procedure for assessing or measuring the presence of any drug. These assays involve growing cells in a controlled environment to assess the effects of food additives, contaminants or the novel ingredients on the cellular health, toxicity and functionality. Cell culture assay are the laboratory techniques used to grow and study cells under controlled conditions, allowing researchers to investigate the cellular behavior, drug responses and the biological processes. These assays can be performed under various cell types, that including the primary cells, immortalized cells, cell lines, stem cells, etc. Common types of cell culture assay include the cytotoxicity assay, proliferation assays and the differentiation assays, which help to evaluate the effects of treatments or conditions on cell viability and its functions. These are essential in drug development, toxicity in food and basic biological researches. Animal models uses animals to study the effects of food components or the contaminants on health and safety. These animal models in the food industry are used to study the effects of food additives, ingredients and the nutritional interventions on the health and safety. They help the researchers assess the impact of new food products on digestion, metabolism, toxicity and the potential allergenicity before human trials. Common animal models such as rats, mice, pigs are selected for their physiological similarities to humans in the digestion and the metabolic processes. These studies are crucial for ensuring the food safety, understanding the nutritional benefits of food and developing the functional foods. Ethical considerations drive efforts to minimize the animal use and find alternatives where possible. Next, coming to the fermentation test. Fermentation tests are used to assess the ability of the microorganisms to ferment specific carbohydrates, which can help in their identification. This is nothing but in food analysis, they are used to assess the ability of the microorganisms such as bacteria and yeast to ferment specific sugars or carbohydrate. These tests helps to identify the microorganisms or the microbial species involved in the food production. such as in dairy, bread and the alcohol beverages, as well as spoilage microorganisms in the food. By monitoring the production of the acids, gases or alcohol, fermentation tests can also evaluate food quality and the safety. They are also used to ensure the proper functioning of fermentation processes and to detect the contamination in fermented food products. Fermentation tests are essential for the quality control in the food and the beverage industry. These biological methods are crucial for ensuring the food safety, quality and the authenticity. They are widely used in regulatory compliance, food industry, quality control research to detect the contaminants, pathogens, allergens and other biological relevant components in the food products. Next is a nutritional questionnaire for the food analysis. In simple way, if you want to say, it is to understand someone's nutrition and the overall health. It is a tool used to gather information regarding the individual's or the group's dietary intake, eating habits and the food preferences. It helps in assessing the nutritional status and identifying potential dietary deficiencies or the excesses and understanding the relationship between the diet and the health. Such questionnaires can be used in the clinical setting and mainly it's used in the research studies and the public health initiatives. In order to research on something, they want to gather some information. Below is the basic structure of the nutritional questionnaire. First of all, they collect the information regarding the demographic information. It includes name, age, gender, height, weight, occupation, physical activity, is they are sedentary or moderate or fully active. Next thing is they collect the information regarding the health and the medical history and the current medical conditions. Do they have the diabetics or hypertension and family history of the chronic diseases, allergies or food intolerant medications or supplements they take currently being taken. And next is the dietary intake, 24 hours dietary recall and all foods and beverages consumed in the past 24 hours, how much they eat, that is the portion size, and how do they cook, that is the cooking methods, and which brand they eat more, that the brand names if applicable only. And food frequency questionnaires, what are the questions they ask in this? The frequency of consumption of various food groups, do they eat daily or weekly or monthly, rarely or never. Questions regarding that, how much often they consume the fruits and vegetables, who eat whole grains and consume dairy products and eat meat, poultry, fish and other alternatives and consume sugary snacks and beverages, and how often do you consume the fried or fatty foods. And next is the portion size estimation. They ask the visual aids or descriptions to help the respondents estimate the portion size, how much they eat. Next is the eating habits. How many times do they eat meals or snacks per day, and do they eat regularly the meals or the meal type patterns, and dining out frequency, do they go to restaurants or fast food centers, and cooking methods preferred, do they eat more the baked foods or frying foods or grilled foods, and consumption of the water and other beverages and use of the nutrition supplements, that is vitamin, minerals and the protein powders. And next is the food preferences and the restrictions. What do they like and dislike of the specific foods or the food groups, and cultural or the religious dietary practices, do they eat vegetarianism or the kosher or halal, and food allergens or the intolerance specific diets followed, and the low carbohydrates and the gluten free. Some will have the milk intolerance. Knowledge and attitude towards healthy eating, towards nutritional awareness of nutritional guidelines, recommended dietary intake of fruits and vegetables, sources of nutritional information, internet, healthcare providers, family, and attitudes towards healthy eating and the nutritional label precisions of personal diet, healthy, balanced and needs improvement, and the lifestyle factors, physical activity level, type of frequency and the duration, and sleep patterns, stress level, smoking and the alcohol consumption. And next is the specific nutrient intake, questions regarding the targeting specific nutrients of interest, calcium, iron, fiber, omega-3 fatty acids intake. Next one is food security and accessibility, access to fresh and affordable food, frequency of grocery shopping, availability of cooking facilities. And the last one is the open-ended questions. Additional comments or concerns about the diet and the nutritional goals related to the diet and health scoring and analysis. After collecting the whole data, The responses can be analyzed to determine the nutrient intake levels and identify dietary patterns, how do they eat, and assess the risk of the nutritional deficiency or excesses. The information can also be used to provide personalized dietary recommendations, create nutritional plans, or guide public health interventions. The nutritional questionnaires can vary in length and detail depending on the purpose of the analysis and the population being studied. They can be administered in person, over the phone, online, or through self-administered paper forms. With this, we have completed all the types of methods for food analysis in brief, that is subjective, objective, and others. Now we are going to study about the classical methods of food analysis in detail, that is water analysis, as analysis, protein, lipid, carbohydrate, dietary fiber, alcohol, pH, and acidity analysis. We will see in the next class. Thank you.